This video demonstrates some features of the ERISA Grameen pan-genome site. Suppose a user is interested in grain size genes, and starts entering the trait term grain size in the search box and then choosing the matching term. The search results show 318 genes across the entire genome. One way to refine the results is to select a region. This region on chromosome 3 filters the results to a list of 9 genes. Notice GS3. It can be entered to pinpoint this grain size related gene. There are 8 active information tabs under this gene. The homology tab opens by default when there is only one matching gene and displays the gene tree related data. In this case, a POACI-specific gene family with 27 members. The alignment overview indicates a G-protein gamma-like domain shared by 5 genes. You can click on tree nodes to expand or collapse a subtree, and resize the slider at the top to zoom in. This tree has diverged from the reference Japonica genome and sorghum and maize orthologs. The red tick marks show exon boundaries, and suggest insertions at the 5th exon in the other 22 orthologs, which are further segregated into roughly 2 subgroups by the position of the 2nd exons. Multiple sequence alignment view further confirms the segregation pattern. Neighborhood conservation view displays 10 genes upstream and downstream of the GS3 gene, color-coded by gene family, and in the center, the GS3 orthologs appear in shades of green, indicating protein sequence similarity. In a perfectly conserved neighborhood, you would observe 21 colored gene families aligned with precise structural order. However, in reality the syntonies are often broken up by evolution, and you would find copy number variation or gray genes from other families that shift the alignments. For example, if you click on this gray gene on Azucena, you can see 10 additional genes in this family, probably arising from structural variation. The Pathway tab imports a pathway diagram from Plant Reactome and focuses in on the formation of RGB1 GS3 complex, a reaction in the regulation of seed size pathway. The Papers tab lists the publications describing the GS3 gene. The XREFs tab lists various names and identifiers assigned to this gene by different organizations and sources. The pan-ERISA pan-gene name is a recent contribution from the pan-ERISA group where they cataloged the genes across the MAGIC 16 rice genomes to pan-gene groups and gave each group a unique standardized identifier. Click on this pan-gene name to bring up the genes in this pan-gene set. The Expression tab imports curated EBI Atlas expression data and provides both anatomical expression maps and heat map views. The Location tab shows the genomic region and gene structure and also serves as an entry point to the Ensemble Genome Browser gene page. The Sequences tab provides means for users to copy the genome transcript or peptide sequences for this gene. And lastly, the Germplasm tab lists accessions that harbor loss of function alleles for this gene and links to stock centers for that germplasm if available.